In this lesson, we'll do a quick review of the reproduction unit up through and including mitosis. We began with an introduction to reproduction. What does it take for a living thing to make more of its kind? Well, it's going to need building materials and information. After all, if the parent is going to make offspring that resemble the parent, there must be some kind of information so that the parent knows how to construct offspring with similar traits as the parent. We call this reproduction with heredity, and it is now understood to be a feature of all living things on Earth. So if a cell can reproduce to make two daughter cells, the cells come from pre-existing cells. By contrast, for a long time, lots of people believed that cells might spontaneously generate. In fact, that idea has ancient origins, but many people for a long time believed that dead meat could spontaneously generate into worms, that broth could spontaneously generate into bacteria, and Pasteur's clever experiments in the 1800s discredited, finally put a nail in the coffin of spontaneous generation. Pasteur argued that bacteria come from pre-existing bacteria. They might be floating in the air, and if they can enter the flask and get to the broth, they will reproduce. If they're trapped, then the broth will remain sterile. But it took time for Pasteur's results to persuade biologists. In fact, some proponents of the cell theory believed that the nucleus itself was somehow spontaneously generating when a cell reproduced to make daughter cells. Why would they believe that? Because the nucleus seemed to disappear prior to cell division and then reappear in the daughters as if it was spontaneously generating. Today's biologists are going to say that the two new nuclei are reassembling, and that what happened was the nuclear material in the parent cell was being duplicated and then delivered to the daughter cells, with two new nuclei being reassembled. Reassembly is different than spontaneous generation. In the late 1800s, scientists using large single-celled organisms like amoeba did a simple but difficult experiment. They would chop the cell in half, or close to it, and tried to make two pieces, one with a nucleus and one without. What they found was that only the piece with the nucleus survived and reproduced. The part without died. And this suggested that the nucleus was necessary. Incidentally, also notice there was no spontaneous generation then in the part that had no nucleus. So from this, then, the question is, well, why is the nucleus necessary? Well, here we see the work of Hammerling helps us understand the importance of the nucleus. He was working with a single-celled algae, acetabularia, had an interesting feature. If you chop off the cap, it can regenerate the cap, and Hammerling suspected that the nucleus was instrumental in that ability for regeneration. Now, Hammerling argued that the nucleus was the repository of information of a kind that controlled processes like regeneration and reproduction. Both regeneration and reproduction require information. If you're going to regenerate a missing part of the cell, you have to have the information to build the right part. Just as in reproduction, to make offspring that have similar traits as the parents, there must be some kind of information that is guiding the construction of the offspring. You'll recall in the two-cut experiment, a piece of stalk with no nucleus can regenerate a cap, and this suggested that the nucleus was delivering important molecules up into the cytoplasm. Later experiments with radioactivity identified them as information molecules, not building materials, not proteins. We call them messenger RNA, and they are temporary recipes to build proteins. Interestingly, the footless stalk never did regenerate another foot. And so we conclude from this that the information to build another nucleus must be in the nucleus itself. So what is in the nucleus? The answer, we learned, is chromosomes. Now here we see a picture of the nucleus of a cell, and inside the nucleus we have a long molecule of DNA that's wrapped around proteins called histones. Together, those two kinds of molecules make up what we call chromosomes. Now we've learned that DNA is the hereditary molecule of the chromosome. It's the one that can store information, be duplicated, and change over time. So we're going to think of chromosomes as hereditary structures, but specifically the DNA molecule itself is the hereditary molecule. Also, we want to make a distinction between DNA, the physical molecule, the double helix, versus the term gene, which is the information stored in the DNA molecule. We would say that DNA stores the information to make proteins, and one recipe to make a protein is called a gene. Now we've learned that Hammerling's information molecules leaving the nucleus are messenger RNA molecules that are going to be built from DNA molecules, and they will be temporary copies of the information stored in DNA. So if a gene is the recipe to build a protein, so too is a messenger RNA molecule. It is a temporary recipe to build a protein. Now, it's just that the DNA molecules are more permanent information molecules, and they're used to build the temporary copies called messenger RNA. Now, the cell uses these chromosomes to build proteins during what we call interphase of the cell cycle. The cell is growing, it's metabolizing glucose, it's building proteins, it's just doing all the chemistry of life. In order for the cell to build proteins using the information in chromosomes, the DNA has to be uncoiled from those packaging proteins, those histones. So the stored information is accessible to the cell's machinery for the purpose of making proteins only when the DNA is uncoiled. Now, importantly, if the cell is going to go on to reproduce, it must duplicate its chromosomes. So cell reproduction requires chromosome duplication, so a new nucleus can be formed in each daughter cell. So this DNA molecule will have to be duplicated, and then the duplicate chromosomes will have to be delivered to daughter cells. That process takes place in a process called mitosis. Mitosis is summarized here in 4 stages: condensing, lineup, separation, and division. So during interphase, the chromosomes are duplicated, but they're still attached. Then they condense during condensing stage, so they appear to be X's. Now they're just sort of randomly arranged here in the nucleus. The nuclear membrane breaks down so that the microtubules can find them and then line them up. And now the duplicates detach, and one set of chromosomes goes one way, the other set goes the other way, and then the cell will divide into 2 cells. A nuclear membrane will reform around the chromosomes, and then the DNA will begin to uncoil again as the cell enters interphase. This allows the cell to access the information stored in DNA. Notice that the nucleus is not spontaneously generating, it is reassembling. The nuclear membrane is reassembling, and then the DNA is uncoiling for use during interphase. So during interphase, the cell is using chromosomes to make proteins. During mitosis, the cell is moving the chromosomes into daughter cells.